Hi everyone, thank you Sorel for the kind introduction, and thank you for inviting me to the Sarcoma UK and all the people who are here to engage today. It's quite interesting because I think I'm the only sort of representative of basic science really, and I think it's us who are perhaps in a way most detached from the community, most away from patients, so it's really a privilege to tell you a little bit about what we do in the lab and what's important for us to learn from you. So a tiny bit more about me. So I really got interested in cancer during sort of third year of my BSc project, where I started working on lung cancers, on one of these targetable mutations that you heard from one of the oncologists earlier in the session. So I worked in the same lab for 2 or so years as a technician. And Lucia, my boss, suggested that I include some images of my life in London and in other places, and all I could really find is images of the lab, which perhaps sadly to some, but not to me, really illustrates what research is about and where you usually spend your time. And I then moved to York, where I worked on kind of more fundamental basic science, not on cancer, but I always knew that I wanted to come back to a problem that is clinical, that has patient applications, not just basic discovery that fulfills our sort of curiosity, but doesn't contribute to well-being of patient populations. And that's how I ended up starting my PhD last year at UCL Cancer Institute. So this is our group so far. Lucia, my boss, in the middle, and Georgina is the research technician who will soon be starting her own PhD in October. And this is where we sit in Huntley Street. This is where the labs are based. So of course we are very fortunate to be well placed in London in a convenient place. And I think what's important to highlight also that we are very lucky to be part of the bigger clinical community of people and of resources, so particularly RNOH, an iconic trial where we can harvest the donations of patient tissues, which enable our research to be more representative and clinically relevant. And two people who have really been leading and enabling this work is Professor Flanagan and Professor Strauss, who help us to ensure that what we do is clinically relevant and ensure that the basic science questions we're asking have some potential to influence clinical guidance, which is what we want to achieve. So you all know this better than I in various different ways, but I think just to introduce the disease and why do we study it, what's the problem? So what we study in our lab is osteosarcoma. So this is a primary bone tumor. So unlike soft tissue sarcomas that you perhaps have heard a little bit more of today, primary bone tumors, as the name suggests, they originate in the bone. And really just to highlight the problem with these tumors, and I think it also alludes to some of the histopathology themes that people have talked about, is that each tumor is different, but within a tumor you have different tumor cells. So not each tumor cell is the same, and you have also non-tumor cells within a mass of cancer cells. Which suggests that maybe those cells, those basic units of a tumor mass, may be behaving differently. And so the way that osteos are treated is by very intense cycles of chemotherapy. So patients undergo 5 cycles of combined chemotherapy of 3 drugs, 2 cycles in a go, after which they receive surgery, and usually it's followed by further adjuvant chemotherapy treatment. And with this very intense cytotoxic regimen, which obviously has massive impacts on quality of life of patients, still about 50% of patients do not reach 5-year survival. So there's a very clear split as to the response in the patient population, and we don't know why that is. And this is where basic scientists come in, or try to come in, to understand how is it that some patients do better than others, and what does it mean at the cellular level, so at the level of the tissue. What we want to understand is why do some tumor cells are able to survive chemotherapy and others are killed. And so particularly for my project, what we're looking at is we think that perhaps in simple terms, as an analogy, there are some tumor cells that are sleeping. So they are not very active. They are able to survive without division. And division is when one cell becomes 2 cells. So that's the fundamental process of tumorigenesis, i.e. establishment of a tumor. And the problem with that, as some of you will know, that chemotherapy, so standard of care in osteosarcoma at least, is a combination of 3 drugs that mostly target cells that are dividing. So the cells that have the ability to become 2 cells from 1 cell. And of course that would create a problem if you have cells that are not dividing. So what we think that such population of cells may exist and therefore be able to survive the chemotherapy treatment. That creates a problem when we withdraw the chemotherapy, when we complete the cycle of chemotherapy, because could it be that those sleeping cells, so the cells here depicted in red, are the cells that then provide the tumor outgrowth, right? So the cells that perhaps lead to relapse or perhaps enable metastatic disease. So ideally, if we're able to understand these, if we're able to identify these cells, if we're able to identify how these cells behave and understand their behavior, we might be able to find new treatments to combine with chemotherapy to ensure that not only the cells that we can normally kill with chemotherapy are killed, but those few remaining cells that may be the most evolved, maybe the most clever, are also killed to eliminate the whole tumor mass, which hopefully would enable us to reduce that dichotomy in patient populations between the good responders and the poor responders. So how do we do that? So what does my day involve? How do we start answering these questions? Well, the first thing we need to do, we need to grow these cells. And when I say grow these cells, so we want to mimic, replicate the process of a tumor growing, and we do that at the cell population. So 1 cell becomes 2 cells and vice versa. And you can see an incubator here full of flasks. So these are plastic dishes, and cells grow on the surface of these plastic dishes. And our daily job is to ensure that our cancer cells are happy, as abnormal as that sounds. And the reason we want them to be happy, because then we want to find ways how we can kill them, right? Yes, so you have to be a particular person to embark on this journey as well. And what I'm showing you here is an example of some cells. So encircled here is 1 cell of osteosarcoma derived from a patient. And this is really important for us to emphasize. So this figure here is a small sample of a tumor tissue that we are very lucky to have. Patients donate to us some samples during resections or biopsies that then we can process, and we can derive cells, so only the cancer cells, from a tumor mass that we can then grow as what we call a cell line. So a population of cells that is more representative of what happens in patients because it's fresh patient material. And really this work has been headed by Georgina, the research technician that I've been telling you about. I'm gonna kill the sound here and just talk you through this. So this is a typical scene on a morning. So you can see the incubators here, and this is our cell culture room. And a typical need for us in the morning is to check our cells. Are our cells happy? Do we have enough cells to do our experiments? Are the cells that are supposed to be dying are dying? And another aspect of work in research in the lab is also training people. And you can see an example of Lucia explaining to one of our students the basic biology, the basic techniques of growing these cells of cell culture. And really I have a couple of students now, and I think it is important to also emphasize that it's a question of maximizing what we can do in the lab by having more hands, but then also passing our knowledge, because as we all know, cancer is not a generational problem, it extends across generations. So a little more examples of my life, my space, how does my life look like? So this is a little bit dishonest, I tidied this up, it's not actually how my table looks like. Life in research is actually quite messy. But really this is where I spend my time. So this is a sterile hood, so it's like a cabinet where we grow our cells in sterile conditions, because we want to make sure that the only things we are growing, we are expanding, right, are the cancer cells. And this is a picture that Lucia has taken when I started my PhD last year, being very excited with my first experiment, and I've been doing a few since then. And really, to wrap it up, I think hopefully I've given a little flavor of what this basic science is about, but to highlight some of the challenges of basic research, and why these challenges prevent us from going forward at a quicker pace. So first of all, with all the gratitude to our funders, because of how expensive things are simply in our economy, in research, in biotech, we are still limited by resources. So those plastic dishes that I told you about, right? I can use them twice a week, right? So a single dish, I get rid of it at the end of the week. Now you can imagine if I have 10 different cell populations in 10 different dishes, right? And a box of them cost £100, for example, right? And I can get through that box in a month. And this is simple, basic resource, right? This is not outrageous experiments where we need to consider, will we have anything left? Time, so in terms of longer scale time, so Sorel mentioned, so my PhD, for example, is 3.5 years, and it's really a minimum of what you can do in research setting, because experiments take so long, because we need to accumulate resources, we need to understand our field. But on the shorter scale of time, right, it's also about understanding, are we doing the right thing? Taking time to ask ourselves, is this the right experiment to perform? Not just be led by curiosity, but be led by the question of, is the experiment I'm doing valuable? Can it contribute knowledge? And the only way we can do that is actually to take ourselves out from the lab and think a little bit, which is counterintuitive to what I want to do as a researcher and as a basic scientist. And lastly, really, it's the unknown of the biology. And I really like the saying that we know the things we don't know, but there are also some unknowns we are not aware of. And I think with these rare diseases, particularly because we're so limited by numbers, and I think this is a theme that has resonated throughout the day. There must be things we don't know about osteosarcoma that prevents us from making progress quicker. And it's really, I think, if we're able to overcome these challenges or fight against the challenges as best as we can, I think this is where the potential of discovery lies. And really, lastly, I think when you give these talks about the research work, the last acknowledgement slide is kind of like what you do, but actually it's quintessential, I think, to my talk and to what we're doing here today, because I've only started in October, and each of these names have either said something to me or shared something with me that has helped me in my project already. And you can see how that amplifies both in scale but in numbers across 4 years. And above all, I wouldn't be here, I wouldn't have the grant, our lab, Lucia's lab, wouldn't have the grant without Sarcoma UK. I wouldn't have my plastic dishes to grow my cells, the cells wouldn't be growing, and maybe some wasted potential would be achieved. And also Sarcoma's UK funding, I think, has been quintessential. It was early funding in the lab that has now actually fueled more funding. So we will be having a new postdoc and another PhD student coming in. And thank you all for listening.